Moving on, we have our next presenter, Saif Rjaibi. Go ahead.
Good morning, everyone. My name is Saif, and I'll be presenting my summer research, which is on an image analysis workflow for characterizing adipocyte lipid droplet expansion in a 3D in vitro obesity model. So the motivation for our research is that obesity has been increasing in prevalence rapidly over the past couple decades. So for instance, it has tripled since 1975, with over 650 million people obese worldwide today. And in certain countries, like the USA, it represents over 40% of the population. And the concern with this is that obesity has been linked to the development of many comorbidities, including diabetes, cardiovascular disease, and cancer. So our current understanding of obesity is that in most cases, it begins with caloric overload. So when we eat, we intake these lipids, and these are stored in adipocytes, which are the lipid storing cells in our body. And under normal conditions, these adipocytes intake the lipids and expand to store them, and then they release these lipids when we need energy, such as during exercise. However, during obesity, because there is a caloric excess, then these adipocytes increase in size and become hypertrophic, which then leads to adipocyte dysfunction and subsequently obesity-associated disease. However, the mechanisms by which adipocyte hypertrophy leads to dysfunction and then subsequently to disease still requires further research. Hence, the McGugan lab has developed a more physiologically relevant 3D in vitro obesity model to study adipocyte dysfunction. And so like mentioned in the previous slide, first these lipids are stored in lipid droplets of adipocytes, and these lipid droplets are numerous and of varying size. However, to mimic the cell hypertrophy caused by excess nutrient intake in obesity, we then stimulate these adipocytes with lipids, and based on previous literature, we believe that these stimulated adipocytes will have bigger lipid droplets, and these lipid droplets will be in lower number due to them expanding and agglomerating as they increase in size. So after developing this model, we would still need to quantitatively prove that we are indeed recapitulating these obesity hallmarks of increasing lipid droplet size. And so this begins with staining. So staining allows us to image our lipid droplets and visualize them under fluorescence confocal microscopy. So we use a DRAC5, which stains the cell nuclei, and the Bodipi stain, which stains lipid droplets. And off on the right here, you can see a video of what our culture looks like, and there are many lipid droplets and cell nuclei. And so, like you may have noticed, the abundance of lipid droplets actually poses a challenge for us to actually quantify and measure the size of these lipid droplets. And so therefore, our goal in this research is to develop a robust and reproducible workflow to count and measure the size of lipid droplets in order to validate our 3D in vitro obesity model. And so we use the Fiji imaging software following a workflow of projecting, filtering, thresholding, processing, and analyzing, which I'll get into the details of each. So projecting transforms the 3D stack that you can sort of see on the left here to a 2D image. And so we do this by using what's called a maximum projection. And so we look at every XY coordinate, and for each XY coordinate, we look at the pixel intensity across every stack in the third dimension, the Z dimension. And if we take the maximum of these pixel intensities, and if we do this for every XY coordinate, then we're able to form what's called a maximum intensity projection, which is in the 2D space, and this is easier for us to work with in the subsequent steps. Next up, we do filtering, which enhances the borders between lipid droplets. And so although the differences may seem slightly subtle between image 2 and image 3, you can sort of see that image 3 is slightly sharper, and this will help our imaging workflow better be able to discriminate between lipid droplets and background. And if you'd like to know more details as well on the mathematics behind filtering, I'd be happy to explain more of that afterwards too. Next up, we move on to thresholding. So during thresholding, we set a threshold, and If a pixel has an intensity above that threshold, it gets rounded up, such as in the light green that you see in figure 4. Or if the pixel intensity is below that threshold, it gets rounded down to the background, which appears in black in the image 4. And so essentially this binarizes our image between the light green that you see and the black, which is the background. So next up, we move on to processing, and this allows us to segment adjacent lipid droplets and remove artifacts. So one thing you might notice from our thresholded image in image 4 is that we actually have lipid droplets that are co-joined together. And that poses a problem because we'd like for our imaging workflow to be able to count each of these 3 here as individual lipid droplets rather than one co-joined lipid droplet. And so to solve that, we use a water shedding tool, which allows us to segment these 3 lipid droplets so we can count them individually. Now, another problem after thresholding is also that we do seem to have certain pixels show up that are above the threshold, as you can see in this zoomed in shot of figure 4. And that also poses a problem because we would like to not include these small pixels here, or just a small number of pixels here, because we wouldn't feel confident that they are indeed lipid droplets. And so to work around that, we use an opening tool, which I'd be happy to describe as well. And that allows us to remove these small pixels to make sure we don't mistakenly count them as lipid droplets. Next up, we can feed that processed image to our imaging software, and it outputs figure 6 here, which shows the contours of all the lipid droplets identified by the system, and each of them are labeled. And so having developed a macro, we can then take this output image and sort every lipid droplet identified into user-defined size ranges. So for instance, there are 130 lipid droplets between 0 to 1 micrometer squared, and so on. And if we add them all up, we can also extract the total count of lipid droplets, or alternatively, represent this information in terms of lipid droplet occupied area. For example, the 130 lipid droplets between 0 to 1 micrometer squared represent 95 micrometers squared in total. We can also get the total area and also represent this in terms of the percentage lipid droplet occupied area for every individual size range. So ideally, we'd use our imaging workflow that we developed to be able to validate our 3D in vitro model and show that there are larger lipid droplets and fewer lipid droplets upon lipid stimulation. However, due to COVID-19 lab closures, that experiment is still ongoing and we still have to obtain those images. But nonetheless, we can still try to validate our workflow by assessing donor-to-donor variation. So for instance, if we look at donor A images, we see that they have a lot more lipid droplets, and these lipid droplets are typically smaller, versus donor B, which has a visually less lipid droplets, but some of these lipid droplets do seem bigger. And so if we actually apply our imaging workflow, we do notice that it appears that donor A has much more area occupied by its smaller lipid droplets compared to donor B, whereas donor B seems to have a lot more area occupied by its larger lipid droplets compared to donor A. And so this gives us some confidence in our imaging workflow because it may be somewhat representative to the images we would obtain upon lipid stimulation. And to address our current limiting steps is that because we are doing a maximum projection, which takes what is inherently a 3D model and transforms it to a 2D space, that does provide a limitation that if certain lipid droplets were one on top of each other, then they would be projected onto the same XY plane. And that's one of our future steps. And so to summarize, we have developed an image analysis workflow, and this will help us validate our 3D in vitro obesity model and make sure that we do obtain larger lipid droplets and that there are fewer of them upon lipid stimulation. And once validated, we hope that this model will serve as a foundation for investigating future therapies in obesity-associated disease. And so with that, I'd like to thank Farah, who's been the graduate student supervising me, Professor McGuigan, and everyone in the McGuigan lab. Thank you.
Thank you, Saif, for your presentation. We now ask judges or participants if they have any questions, please to turn on their mics.
Thank you very much. That was a great presentation. I have one quick question regarding the plan for 3D printing. I was wondering if, like, what would be the material that you're gonna use it for the lipids. Also, I didn't quite get what software did you use for the image processing and the spacing between, because it's like 3D, right? So in Z direction, what is the interspacing layer of like your proposed plan?
Yeah, sure. So I guess just to address the many questions, so the software that we're using is the Fiji image analysis software. In terms of, I think you asked the question for maximum projections, I think. So I guess I'll just go back to that slide here. Just to clarify your question, were you asking like what the say like differences in the slice dimensions were?
Yeah, well, what is the, like in Z direction in each layer, what is the distance?
Yeah, for sure. So that depends on our fluorescence confocal microscopy. So I believe they are on the range of microns. And so essentially based on how we use our fluorescence confocal microscope, the microscope essentially makes different slices as it goes through the 3D culture, and then we essentially get many slices. Usually there are around 40 to 70 slices, and then we look at each slice and take the maximum intensity across each of those slices for every XY coordinate.
And sorry, my last question was like, what would be the material that you are gonna like build with the 3D printing?
Yes, so we're not doing any like 3D printing of sorts, but we are using a hydrogel, which is where we are culturing our adipocytes in.
Great, thank you very much.
If there are any more questions, I ask that participants or judges raise their hand or type something in the chat. All right. Thank you so much, Saif, for your presentation.
Thank you.